Hello, welcome back to this channel. Today the topic of our video is bacterial staining. That is an important topic in microbiology. As we all know, human eye cannot see microorganisms, due to which we need to magnify the size of microorganisms to see them. For that purpose we need a magnifier, that is called microscope, which enlarges these microorganisms to the size of 1,000 times than their original size. The size of bacteria is measured by micrometer, and the size of most of the bacteria ranges from 1 to 3 micrometer, out of which the smallest bacteria is Mycoplasma, that is a wall-less bacteria, and its size is 0.2 micrometer, whereas the largest bacteria, that is Borrelia, is 10 micrometer in size. So when we are watching the microorganisms under microscope, we need to stain them to differentiate between the microorganisms. According to the pH, all stains are either acidic, alkali or neutral. Generally, the acidic materials are stained with alkali, while in contrast the alkali materials are stained with acidic stains. We classify the staining methods into the following categories that include simple staining, differential staining, example is gram staining, acid-fast staining, negative staining, flagella staining, capsule staining, and spore staining. First, we will discuss simple staining. In simple staining, we use only a single dye that can be either methylene, blue, carbolfuscin, or safranine. In this staining, we just watch the shape of organisms, whether it's rod, cocci, or spirochetes. The procedure of simple staining is, first we make a thin smear of the given sample on the glass slide, and then it is air dried, and then it is covered with methylene blue, and then it is washed with tap water, and then again air dried. After drying the slide, it is observed under the microscope. If the given sample is bacteria, then we can see either bacilli, cocci, or spirochetes. So this picture shows the simple staining technique. In this picture, it is stained with safranine dye that shows the red or pink colored bacteria, and in this picture it is stained with methylene blue, and blue color of bacteria is seen. In this simple staining we only can see the shape of bacteria. We cannot differentiate either it's gram positive or negative. So next method is differential staining. Example is gram staining, and it is the most important staining procedure that differentiate the bacteria into two main categories, that are gram positive and gram negative bacteria. This method was discovered by Hans Christian Gram in Germany in 1884. In this method, we use more than one type of dye, which include crystal violet and safranine. Basically, the mechanism of this staining is based on the differences of gram positive and gram negative bacteria in their cell walls. Gram negative bacteria have a thin peptidoglycan covered by outer lipid containing membrane, whereas gram positive bacteria have a thick peptidoglycan and there is no outer membrane in gram positive bacteria. This picture shows gram negative bacteria in which there is outer lipid containing membrane and peptidoglycan is thin layered, while in gram positive there is no outer membrane and peptidoglycan is a thick layer. Again, it shows an outer membrane in gram negative and a thin layered peptidoglycan, while a thick layered peptidoglycan in gram positive and no outer membrane. This is again the repetition gram-positive with a thick peptidoglycan layer with no outer membrane, and gram-negative with a thin peptidoglycan layer and an outer membrane that contains lipid. What is the procedure of gram staining? In this procedure, a smear of bacterial specimen is made on a glass slide as described earlier, and then it is air dried and then covered with crystal violet for 2 to 3 minutes, and then washed with tap water and then covered with grams iodine for 2 minutes, again washed with tap water and then covered with grams alcohol for 1 minute, and then washed with water and then covered last dye, that is safranine, for 2 minutes, and then washed with water and then observed under the microscope. Under microscope, then we will see either purple colored or pink red colored bacteria. Purple colored bacteria would be called gram positive bacteria, while pink colored bacteria would be named as gram negative bacteria. So this slide shows the procedure of gram staining. In the first step, we will add crystal violet dye, that is a purple colored dye that would stain all the cells to be purple colored, purple or blue colored. In the next step, we will add Lugel's iodine that would act as mordant, and it would bind with crystal violet peptidoglycan complex. And in the third step, we will add alcohol that would act as decolorizer, and it would decolorize the gram negative cells, while gram positive would be stained purple or blue colored. In last step, we would add a safranine red dye that would stain uh, gram negative cells as pink red, while gram positive would be stained blue purple. After making the slide, we will see these gram positive cells that are blue or purple colored under microscope, and these are gram negative that would be pink or red colored. Next staining procedure is acid fast staining, that is also called Ziehl-Neelsen staining, that is named after the pathologist that discovered it. It is an important staining procedure to identify acid fast organisms, the example of which is Mycobacteria tuberculosis. The difference of Mycobacteria from other bacteria is it contains an outer membrane in addition to peptidoglycan or envelope that contains large amount of glycolipids including mycolic acids. This picture shows the cell wall of Mycobacterium tuberculosis. Here this is the peptidoglycan layer, and outer to this peptidoglycan there is an outer envelope that contains this mycolic acid. Uh, Due to this mycolic acid lipid layer, the gram staining dyes cannot penetrate into the cell wall of this bacteria, due to which this bacteria cannot be stained by gram staining techniques. For that purpose, we need to stain it with acid fast staining technique. Procedure of this acid fast staining technique include: again, a smear of sputum is made on the glass slide, and then it is fixed with ethanol and then air dried, and then it is covered with carbamazepine dye and then heated for 5 minutes, and then washed with tap water and then covered with acid alcohol for 1 to 2 minutes, again washed with tap water and then covered with methylene blue for 2 minutes. And then after washing the slides and after air drying, this slide is observed under the microscope to see the Mycobacterium tuberculosis bacteria that would be seen as red rods. This procedure, first step is the primary stain would be carbamazepine that is applied for 30 seconds, and then it is fixed with heat, and then a decolorizer, that is acid alcohol, is added for 20 seconds. That would decolorize other bacteria that are not acid fast, while acid fast would be stained as pink red. Then in the last step, a counter stain, that is methylene blue, is added that would counter stain these non-acid fast bacteria as blue, while acid fast would be stained as pink red. So this slide shows the acid fast staining that shows this pink rods as Mycobacteria tuberculosis or acid fast bacteria. Next type of staining is negative staining or Indian ink. This is a method that is used in diagnostic microscopy. In this technique, the background of the slide is stained while the actual specimen would be untouched, and that would be visible then. In this procedure, a smear is made from the given sample with negrosine or Congo red dye, and then it is air dried and then observed under a bright field microscope. The background would take the color, while bacteria would be bright, and they would be visible. It is used for spirelia for electron microscopy. This picture shows the negative staining of spirelia, where the background is dark in color while the bacteria or spiral shaped spiral bacteria are brighter, and therefore they are visible on dark background. Here again the dark background and the spiral shaped bacteria that are brighter in color. Next staining type is flagella staining, that allows the observation of bacterial flagella under microscope. They are too thin to be seen, so they must be stained with a special dye to be seen under the microscope. Capsule staining. Capsule is basically a gelatinous outer layer secreted by bacterial cells that surrounds and adheres to the cell wall. The main purpose of capsule staining is basically to distinguish between the capsular material and the bacterial cell. Capsules are basically composed of polysaccharides, but most are also composed of polypeptides. Capsule staining employs an acidic stain and a basic stain to detect capsule production cells. Procedure of this staining technique include: first, a small drop of negative stain, that can be Indian ink, Congo red, negrosine or eosine, is placed on the slide, and then a loop full of bacterial culture is placed on the slide to make the smear, and then with another slide is dragged on this ink cell mixture to make a thin film, and then it is placed for and air dried for 5 to 7 minutes. Then this slide is covered with crystal violet stain for about 1 minute, and then it is covered with copper sulfate, and then the crystal violet is drained by tilting the slide at an angle of 45 degree and letting the stain run off until it air dried. Then it is examined and observed under the microscope for the presence of uncapsulated bacteria that would be indicated. The capsule would be indicated by clear zones surrounding the cells. This is the procedure of capsule staining. In the first step, a negative stain is added on the slide, and then with the inoculation loop, bacterial culture is added on the slide, and a heavy smear is made, and then it is thinned out with the help of another slide, and then it is after air drying, it is covered or flooded with crystal violet dye, and then copper sulfate is added on the slide, and then it is blot dried, and then observed under microscope. The bacterial cells would be dark blue in color, while capsule would be surrounding the cells, and that would be blue violet in color. So this picture shows the slide of capsule staining under the microscope in oil immersion lens. These are the bacteria in blue red color, and these hollow zones surrounding the bacteria are the capsules in each bacteria. Next staining technique is spore staining, that is a differential stain used to visualize bacterial endospores. Endospores are formed by few genera of bacteria. Examples are Bacilli. By forming spores, bacteria survive under hostile conditions because spores are resistant to heat, dryness, chemicals, and radiations. Procedure of this staining technique include: first a smear of organism is made on the slide, and then it is heat fixed, and then a small piece of blotting paper is placed over the smear, and then the slide is placed on a wire gauze on a ring stand, and it is heated gently till it starts to evaporate, either by putting it on the staining rack that has been placed over a boiling water bath or via Bunsen burner. Then it is removed from heat and reheated as needed to keep the slide steaming for about 3 to 5 minutes. As the paper begins to dry, then a drop or two of malachite green is added to keep it moist. Then a blotting paper is removed and the slide is allowed to cool down at room temperature for 2 minutes, and then it is washed with tap water to wash the malachite greens from the slide, and then the slide is stained with the saffronin for 2 minutes, and then the slide is washed from both sides to remove the secondary stain, and then the slide is air dried. First a smear is made, and then a blotting paper is added on the smear, and then a malachite green stain is added, and it is placed on the heat for 3 to 5 minutes, and then the paper is removed and it is stained with the saffron dye, and then air dried and seen under the microscope. So this slide shows the spore staining, in which the vegetative forms that appear pink colored while the spore forms are greenish in color, greenish or blue in colors. Thank you for watching this video. Stay tuned to our channel for watching more informative videos like this.